Welcome viewers. Today we will be discussing on the important topic called bacterial transformation. Transformation in bacteria is supposed to be one of the important discoveries in the field of molecular and microbial genetics, as this led to the development of DNA as the genetic material. Actually, in bacteria, there are three different ways of gene transfer. Number one, say for example, is conjugation, wherein the donor cell has got a plasmid which is transferred by the method called conjugation onto the recipient bacteria. And during this process, the recipient bacteria receives the plasmid and one copy is retained within the donor cell as well. The second method of gene transfer is called via transduction, wherein we can see that the bacteriophages that can infect bacteria, they infect the bacteria and they introduce their DNA inside the bacterial cell. And when they replicate and come out of the bacteria during lysis, what happens that sometimes they package the genomes or fragmented genomes of the bacterial DNA. And in the second course of their infection, when they reinfect another bacteria, this bacterial DNA is transferred via the bacteriophages onto a new recipient bacteria. The result of this type of gene transfer is recombination and generation of new traits within the bacteria. Today's topic of discussion is transformation. In this concept, we will see that the foreign DNA gets inside the bacterial cell and the DNA is in the naked state. The DNA can be obtained by the bacteria by three different means. It can be that the dying bacteria is donating its DNA, which is being received by the bacterial cell. Sometimes free DNA is available in the environment that can also be uptaken by the recipient bacteria. And in some cases we have seen that living bacteria secrete some DNA and that DNA is uptaken by a recipient bacteria. The concept of transformation has really changed the entire scenario. So let's have a look that how they take up the DNA, that some bacteria can take up fragments of DNA from external medium, as I said, and the sources of DNA can be DNA from the same species of bacteria, and in some cases we see that they are procuring DNA from different species of bacteria as well. As I said, in some cases we can see that the result of this transfer is that the bacteria procures certain new traits.
Ma'am, what are the probable fates of the foreign DNA when it gets inside the bacterial cell?
Yes, rightly said that the DNA that is getting entry inside the bacterial cell can have different fates. One fate is that it may get integrated within the bacterial genomic DNA. Then the trait that it is carrying will be transmitted to the daughter cells. On the other hand, sometimes the foreign DNA that is getting inside may circularize and may reside as an independent, also autonomously replicating plasmid. Under such circumstances, this foreign DNA will have its independent existence. And in some cases, we have seen that neither of the steps are taken and the DNA gets degraded and it is lost. Such an instance won't be considered as a transformation because we consider only those cells as the transformed cells wherein we can see that there is some change in genotype is occurring, and that change is giving us an indication that this particular cell got transformed. So, we will be starting our journey of transformation by paying tribute to this eminent scientist Frederick Griffith. In the year 1928, he actually established the concept of transforming principle. He did a very simple but interesting and intelligent experiment. Let's have a look into the experiment. He started working with Streptococcus pneumoniae. He has taken two different strains of pneumonia. One was the pathogenic strain and that was giving smooth colonies because of the presence of certain polysaccharides. And the second strain was the rough strain which was devoid of that polysaccharides. The smooth strain was pathogenic and when that strain was injected into the mice died. And the second strain, that was rough strain, when he injected onto the mice was surviving. And the third was he heat killed the pathogenic bacteria, means the smooth strain was heat killed and it was used to inject the mice, and the mice survived. And the fourth experiment was most interesting one, wherein he mixed the heat-killed smooth or virulent strain along with the rough strain. And ideally it should be non-pathogenic to the mice, but he could see that the mice is dying. Then he could conclude or draw inference from the experiment that definitely this heat-killed smooth or virulent bacteria must have transferred something which he called as the transforming principle onto the rough non-virulent bacterial cells. So he was able to define that principle as transforming principle, but the exact nature of that transforming principle was not being unraveled by Griffith. Then many years passed by, and in the year 1944, we could see that Oswalt T. Avery, Colin M. MacLeod and Maclean McCarty, these three scientists took a vital role in establishing the exact nature of this transforming principle. What they did, they treated this transforming principle once with a lipase which will destroy any lipids present in it. And they could see that the same experiment, when repeated, gave the exact result that Griffith got. Therefrom, they could infer that the transforming principle was not definitely a lipid. Similarly, they treated the transforming principle with a protease, and they got the similar results that the transforming principle was still having its virulence in it. Thereby, they could infer that it is not the protein. Finally, when they treated the transforming principle with the DNAs, then they could see that the transforming principle has lost its virulence. And that was supposed to be the most remarkable discovery in the era of molecular biology, wherein it was established that DNA is supposed to be the genetic material, it was supposed to be the transforming principle. Therefrom, the journey emerged, and henceforward the transformation is supposed to be the most remarkable discovery in the field of molecular and microbial genetics. After that, the doors were opened and the molecular mechanism of transformation, conjugation and transduction was unraveled. So, if I try to focus on transformation, you must be thinking of that what are the major applications of the study of transformation. So transformation was supposed to be most important in mapping of the bacterial genomes, because scientists could find that there are certain cases wherein conjugation cannot be used to map the bacterial genome. Under such circumstances, transformation was used for carrying on the mapping of the genomes. And if I look into the recombinant DNA technologist's point of view, then we will see that transformation is supposed to be one of the easiest method of introducing foreign DNA inside any bacteria. Initially, transformation was discovered in case of bacteria. Later on, transformation mechanisms in plant cells and animal cells were also discovered. So, if I consider transformation, then the ability of the bacteria to uptake DNA or to procure the capability of transformation is termed as competence. This competence is an acquired phenomena by the bacteria, right? So certain molecular operons are switched on to execute the function of transformation. So if I broadly categorize the transformation, they can be divided into two categories. Number one is natural transformation, means there are certain bacteria residing in the environment that they are capable of uptaking DNA on their own. Such examples are Bacillus subtilis, Streptococcus pneumoniae, Haemophilus influenzae, Neisseria gonorrhea and so on. On the other hand, there are certain bacteria which are not naturally transformable and we make them competent by artificial means like application of calcium chloride or heat shock, or by using electric current or electric shocks, we transform not naturally transformable bacteria with a foreign DNA. So this is called as engineered transformation, and the most promising example is Escherichia coli. So let's have a look on what is happening during bacterial transformation. This picture clearly signifies that the DNA which is present in the environment, it exists as a double-stranded DNA and it binds to the outer cell membrane, and when it passes through the cell, it gets converted into a single-stranded DNA. So the DNA that is getting entry inside the bacterial cell is getting as an single-stranded DNA. And after that, as I said, it can have different fates. Either it can get integrated into the chromosome and it will be transmitted onto the daughter cells. On the other hand, it may just remain as an independent entity and it will be chopped up by the nucleases present in the bacteria. So, if I try to focus on gram-negative bacteria, first step is binding of the double-stranded DNA to the outer cell surface, then movement of the double-stranded DNA across the outer membrane and cell wall, then degradation of one of the strands, and finally translocation of the remaining single-stranded DNA into the cytoplasm of the cell. There are at least two stages of interaction between the foreign DNA and the bacterial cell or the recipient bacteria. The initial stages of interaction between the DNA and the bacteria is a reversible interaction, and during this interaction, this DNA is sensitive towards cleavage by the DNase enzyme. And the second stage of interaction is one when the exogenous or the foreign DNA gets entry inside the bacterial cell, and this stage is supposed to be insensitive towards any interaction with the enzyme called DNase.
How can we estimate the efficiency of DNA uptake?
Yes, this is supposed to be one of the most crucial questions, as while you are carrying on transformation, you really need to calculate the transformation efficiency. In this regard, they have performed an experiment wherein they radio-labeled the foreign DNA with gamma p32, or the DNA was labeled with p32, means phosphorus 32 isotope. And these cells were allowed to infect the bacteria, and after say 15 minutes of time, what they have done, they have centrifuged the cells or acid precipitated the cells, and the cells bearing only the foreign DNA were taken, and the amount of radioactivity incorporated inside the bacterial cells were calculated. And prior to this experiment, what they have done, they have removed all the DNA which have not infected the bacterial cells with the help of DNases, and they were present in the supernatant and were discarded. So, by having a ratio of the amount of radioactivity that has entered inside the bacterial cell and the total amount of radioactivity which was present in the DNA that was infecting the cell, that gave us an idea of the efficiency of transformation. Means how many cells got transformed was calculated in this fashion. Next is, whenever they worked on this transformation experiment, there was a query in their mind that when initial binding of the DNA with the cells takes place, is this a sequence specific binding or this is a non-specific binding? In this regard, what they did, they started their experiment with a Streptococcus pneumoniae cells which were penicillin sensitive, and they tried to transform it with a DNA isolated from a penicillin resistant Streptomyces cells. And what they did, that definite amount of Streptomyces resistant DNA was used to transfer definite number of Streptomycin sensitive pneumoniae cells. And what they did, they increased the amount of calf thymus DNA along with the foreign DNA. It means this calf thymus DNA was used as a competitive inhibitor in the transformation experiment. And as expected, they could find that the transformation efficiency was decreasing as they increased the amount of calf thymus DNA. This experiment helped them to infer that during the initial binding of the DNA onto the bacterial cell surface, it doesn't require any specific DNA sequences. So this thing was proved in case of Bacillus subtilis as well as in case of Streptococcus pneumoniae. And there were certain exceptions in this experiment, that they could find that bacteria like Neisseria gonorrhea has got specificity during DNA uptake. It means if such experiment was repeated with Neisseria gonorrhea, they could find that calf thymus DNA cannot compete with the DNA from the Neisseria gonorrhea cells. From there, they could find that there exists a 10 nucleotide sequence in the Neisseria genome, which is necessary for initial binding onto the Neisseria cell surface. So they could easily group naturally transformable bacteria into two categories. One of them that can uptake any type of DNA, and other class that can take only self DNA. Right? Next question that was hunting in their mind, that whether the initial binding of the DNA onto the bacterial cell surface needs a double-stranded DNA or a single-stranded DNA. To justify this, what they did, they performed a similar experiment wherein that Streptococcus pneumoniae cells which were sensitive towards penicillin were transformed with Streptococcus pneumoniae DNA containing penicillin resistant DNA was there, and along with it they added denatured calf thymus DNA. And they could find that unlike the previous experiment, denatured calf thymus DNA is unable to competitively inhibit the transformation process. So this helped them to infer that during the transformation experiment, it is supposed to be only the double-stranded DNA that binds onto the cell surface. And this was further proved when they renatured the calf thymus DNA, and it was again converted into a double-stranded calf thymus DNA, and they could again see that renatured double-stranded calf thymus DNA is decreasing the transformation efficiency. So this is how that simpler experiments were performed to establish the actual phenomena of transfer of DNA from external sources onto the inner side of the bacteria. Next thing that was very controversial on those days, that people were thinking that what are the stages of DNA uptake? Means what is the fate of the DNA while it is traveling from the outer cell surface onto the inner part of the bacteria. To perform this or to establish this, they performed an experiment wherein they started working with a bacteria which was arginine minus, means it is unable to synthesize arginine. And such a bacteria was transformed with the DNA which is arginine plus. And a kinetic study was done. Then what they did, they allowed the cell and the DNA to remain in contact for say 1 minute, 2 minutes and 15 minutes. And at this time points, the reactions were stopped and the reaction mix containing the cell and the DNA was divided into two halves. Means the cell that have received the DNA was divided into two halves. One half was plated on a media devoid of arginine, and from the other half they have isolated the DNA, and that DNA was used to retransform another class of cells which were again arginine minus. So what they could see, that when they tried to perform this type of exercise at time point 1 minute, they could see they could neither find any colony on arginine minus plates. It means transformation or for initial entry of the DNA, 1 minute is insufficient. Then when they isolated the similar experiment with time point 2, there they could find that they are getting colonies on the plate which is devoid of arginine. That indicated that the bacterial cell have received the DNA. But at this point, when they isolated the DNA and tried to retransform the arginine minus cells, they find no transformants. So here from they could infer that the DNA got entry inside the bacterial cell, but it is still in a non-transformable state. But when they isolated the samples after 15 minutes of incubation with the bacteria, then they could see that they are not only getting colonies on arginine minus plates, But when they re-isolated DNA from such cells and tried to retransform another arginine minus bacteria, they got sufficient number of transformants. So they could infer out that initial entry of the DNA inside the bacterial cell needs at least 2 minutes. But the time gap between 2 minutes and 15 minutes is the time where the DNA remains in a state called eclipse state. In this state, the DNA has got entry inside the bacterial cell, but it has not yet integrated itself within the bacterial genome. And this was the reason that in the 2 minutes time point case, they couldn't find any DNA that could be transformed onto another arginine minus bacteria. So this vital experiment helps us to understand that initial entry of the DNA needs 2 minutes, but to integrate inside the genome, it needs at least 15 minutes. During transformation, it is only the single strand of DNA that gets entry inside the bacterial cell. So to establish this thing experimentally, another small experiment was done wherein they started working with a radiolabeled DNA, and it was used to transform a bacterial cell that was grown in presence of cold DNA, means normal DNA without radioactivity. They could find that the incorporation of amount of radioactivity was 50% of the starting material. And this experiment helped them to infer that it is only the single strand of the DNA that is getting entry inside the bacterial cell.
Ma'am, during transformation, does the DNA undergo addition or substitution?
Yes, this was supposed to be the most challenging question in presence of the molecular geneticists. What they were thinking of, whether the DNA is getting substituted or addition. For that, they performed reciprocal crosses. What do you understand by reciprocal cross? It means that they started working with an X minus strain, and it was transformed with a DNA containing X plus DNA. And they calculated the number of cells that turned out to be X plus. And similarly, this X plus cells, which turned out to be X plus, were again used to transform another lot of cells, and they could find that the transformation efficiency from X minus to X plus is equivalent to the number of transformants from X plus to X minus. And this can only be justified if it is substitution. If it would have been an addition, then these results will never be obtained. Another example that they have taken is that the donor DNA that they have chosen, they started working with two markers. The donor was capital M and small n, and the recipient was small m and capital N. And the transformants were selected for capital M phenotype. And they were getting, either they should get capital M, small n, or capital M and capital N. It means the proportion of getting capital M, small n and capital M, capital N should be equal if it is substitution. On the other hand, if it would have been addition, then in both the cases, all the cells should be capital M and capital N. So they could actually get equivalent amounts of capital M, small n and capital M, capital N. That indicated that it is nothing but substitution. So addition was ruled out by performing these two experiments. Now the question arises that how a single-stranded DNA gets substituted with the double-stranded genomic DNA. So what was proposed here, that certain single-stranded proteins cover up the DNA, and the Rec-A protein will bind to the strand which will participate in the event of substitution. And you can see here synapsis and assimilation takes place, which is followed by nicking of the recipient strand. The recipient strand undergoes nicking, followed by branch migration and trimming. This leads to the formation of a new strand, that the new strand is replacing the older one, and there can again be two fates, that there can be certain cells wherein this new genotype was getting preference. As most of the time we gave such a selection pressure so that the newer donor genotype is being selected out. and in certain cases these cells don't use their repair mechanism, or their repair mechanism was damaged, as a result of which we could see the generation of new transformants or new recombinants were generated.